part of this here. So there are different alleles are different versions of a gene. Most are diploid in nature, where you have two different versions. You can get one from one parent, one from the other. And then most things that appear stark white in nature are homozygous recessive. And so we haven't talked a lot about albinism, but we're going to get into that. It's one of those very rare conditions. A lot of times it shows up on Hardy-Weinberg questions. And it's one of those things where you got to like take the decimal out like 6 places, or you're going to have trouble, because the average person with albinism is like a 1 in 20,000 chance, something like that. It's really rare. So, but that's one of the things they use because it's something we're commonly familiar with. So in the first part of the notes here, it talks about phenotypic polymorphisms. So phenotypes are the expression, the way something looks. Polymorphisms means just different manifestations of that. So if you skip down to part C, phenotypic polymorphism, many versions or types of a gene, they give you an example of blood typing. So we talked about blood typing. You could have the A antigen, the B antigen, you could have A and B antigen, you could have neither one of those and have O blood type. So there's many different types of this one gene for blood, where you have multiple alleles that are coding for different antigens that go on the different blood types. Okay, and then another example where things don't just follow the diploid either or, like your tall or short pea plants, is genotypic polymorphisms. So genotypic meaning the genes, and polymorphism meaning many. So these are many different genes that are causing some type of quality that you see or characteristic or trait. The example that they give is skin color. And so skin color we've talked a lot about. It's easy to see in people these variations. You rarely see anybody that has the exact same skin tone as someone else. And so across the board we see lots of differences there. So let's read through that. I'm going to let y'all split it up. Walker, read that first part on skin color, and then we'll let somebody else read the second part.
Skin color is basically based upon how many dominant alleles you inherit from your parents. The dominant allele makes the protein pigment found in skin called melanin. Every human makes this protein pigment, except people suffering from albinism, a genetic condition where the individual inherited 0 dominant alleles and therefore does not have a single DNA blueprint for making the protein pigment.
All right, okay, work from there.
So they appear white because they have 0 pigment in their cells. In those individuals who did inherit at least one dominant allele, they'll have a skin pigmentation. So basically, the more dominant alleles that are inherited, the darker the skin will be. Race is a man-made construct that tries to group individuals based on geographic origin or degree of skin pigmentation, but we're all human. We now recognize the direct relationship between the environment and the degree of pigmentation. Populations around the equator are darker in an attempt to counteract the harmful effects of sunlight. And as we move away from the equator, the amount of pigmentation decreases generally. Race is an antiquated term. As we now scientifically understand, we are all Homo sapiens. Racism is showing your scientific illiteracy.
Yeah, so I know we've had a lot of racial issues come up recently, especially in the past 5 years or 6 years or so. And the author wrote this way before then, and so, or the notes. And so he's kind of taking a dig at racism, basically saying that, we are all 99.99% the same. There's very little difference in our nucleotide diversity, which is down there under part 3. And so we're very, very similar. I know that there are manifestations and we have different skin tones, and we have different hair colors and eye colors. And so these things show up a lot in humans that you may not see in other animals. Like if you look at chimpanzees, like most chimpanzees look about the same, right? There's not a lot of variation, not hair color, eye color, or even, height and weight and stuff like that. They're very similar. But we see lots of differences. And one thing that they talk about here is skin color. And so there's lots of genes that go into making the skin color that we have. Yes?
So wait, what happens if with the arthropathy diseases? Well, that's when they have the white and
Oh, there's patchy. A lot of times you see a black person with patchy, lighter skin. Is that, it's either impetigo or vitiligo. I can't remember. Vitiligo, yeah. And that would be like, for some reason, those cells aren't producing pigment there. I don't know what's causing that. We'll have to look that up. Maybe you should look that up for extra credit. But that's not uncommon, yeah. And so where that pigmentation is, for some reason, the melanin's not producing stuff, the pigmentation in that skin. And pigmentation varies for everyone that has melanocytes. And so even if you're very fair skin, you have the ability to produce more melanin. If you have very dark skin, you have the ability to produce more melanin. And so this varies among, different groups dependent upon, your melanin production varies dependent upon what? Your genetics, obviously, but then it also depends on the sun. And so we talked back in the hormone chapter, we talked about melatonin, we talked about your tanning response, where the more you're out and exposed to UV radiation, the more your body senses that and the more melanin it produces, so that you're able to kind of block as much of that UV radiation as possible. And that, why are you not just producing the maximum amount of melanin you can all year long?
Because in colder environments, you want to be able to absorb sunlight.
Yeah, I mean, when there's not as much sun, you want to be able to absorb sunlight. Sunlight absorption, we know, is important in the production of. What does your skin produce? Your skin makes its own vitamin D. So I think it's primarily due to exposure to UVB rays. There's UVB, there's UVA, UVC. UVC and B are the bad ones. They're mostly blocked out by the ozone layer. But the UVB rays, I think that's what I read recently, are the ones that are responsible for that vitamin D production. We also talked about maybe its possible role in, the immune system and fighting COVID. And so we talked about that a little bit a while ago. I don't know. I saw a study recently that said that there was no real difference between people with more or less vitamin D in terms of their ability to fight off COVID. So we've seen a lot of up and down and up in the air, and I think it'll probably take a while to actually figure out what the real answer is there. So anyway, and typically what the author's saying here is if you go near the equator, people that are from or have ancestors near the equator, or if they live near the equator, they're generally going to have darker skin because you're going to be exposed to more UV light, right? And so if you think of people in Central America, or in Central Africa, Southeast Asia, along the equator, like Southern India, or Northern Australia, like people all throughout those regions have darker skin tone. And if you think about people from like Northern Europe, you're going to have much lighter skin tone. You don't need as much pigmentation. You need to absorb more sunlight for vitamin D production. There's no need to produce pigmentation if you're not exposed to a lot of UV light, because then your body's actually making extra proteins and stuff it's wasting energy on, right? So, and so it all just depends on your ancestral evolutionary pattern. So regardless of I'll get into this real quick. Regardless of your thoughts on where people came from and how we started out, in terms of evolution, if you started out with, just a few individuals, obviously we have a wide range of individuals now. So depending, whatever your perspective is, then we went from a few individuals, humans, to a lot of different individuals. And there are lots of differences that we see among groups from different places all over the world. And so I think that's something important to consider. That's a good example of how microevolution can change a species very quickly, and so just something that's very evident for us to see even in a very short period of time, okay? And I was like, they're wrong for that. I was like, that is in my notes. All right, so if we skip down to geographic variation within species. So like we just, well, actually, hold on. Measuring variation on a cellular level, gene diversity. So there's lots of gene diversity. It's been documented in humans that most traits are homozygous in nature. There's more homozygous traits than heterozygous. For whatever reason, I guess those like recessive traits are maybe better, or the homozygous dominant traits are wind up being a better trait or a stronger trait, and so that shows up more often. We are going to talk about one where heterozygous is advantageous in a second and look into the details of that. And then skip down to, we talked about nucleotide diversity, 99.99% of similarities between us, right? So we're all very, very similar. There's just a few slight differences that give us the differences that you see. What is a single nucleotide polymorphism, Mason?
This term refers to genes where a single nucleotide difference in sequence can affect the phenotypic outcome. For example, the color of the actual protein pigment associated with normal red blood cell versus sickle red blood cell, a point mutation.
Yeah, so we talked about this single nucleotide polymorphism. This is where you have one change and it affects, it morphs into something and causes multiple changes. Like with sickle cell, not only do you have sickle-shaped cells, you also have a less of ability to transport oxygen. You have you have a possible resistance to malaria. So there are a number of things that come from this one mutation, this one change in a nucleotide. Okay, let's skip down to geographic variation. Differences between populations because each population exists in a different environment. So remember, the environment drives the evolution and change in a species over time. Okay, so cline. Think incline or decline. This term refers to variation within a species that is directly due to an increase or decrease in elevation or latitude. Now, what happens when you go up in latitude or you go up in elevation, Matthew? In general. Does the air get thinner when you go up north?
Higher. When you go north, it's colder.
Colder, right? So as you go up in latitude or you go up in elevation, generally things get colder. So what variation do you typically think of happening when it gets colder to species?
Lighter fur colors.
Well, that's a good one. Maybe lighter fur colors as you got more snow exposure. What else? We talked about lighter skin color definitely going up. I don't know that skin color probably wouldn't happen though. Skin color is different though, right? So that would be probably different for us. If you think about people in a lot of mountain communities like the Himalayas and stuff, a lot of times there is a little bit darker skin tone because a lot of times that UV radiation there can chap the skin. Or if you're exposed to snow, we just talked about that, where the more snow and reflective UV radiation could cause you to burn. So maybe the benefit there would be a little bit darker skin, even though it is colder. So in those higher latitudes. What else? We got, let's just think about like rats and mice and deer and stuff like that. As you get colder, what happens? Thicker fur and probably more fat, right? The ability to, because you want to, if you go deer hunting up north, so I'm a big deer hunter, like down here, our deer are pretty thin on the fat. Like you skin a deer, there's not much of a fat layer. If you go up north, you're talking about a thick fat layer usually under that skin because they're having to deal with temperatures that can be in the minus 10, 20, 30 below at times. So they have to be able to survive that. So typically that's what you see as you get away from the equator or you go up in elevation to survive those colder conditions, especially when you're talking about endotherms like us where you have to maintain a stable body temperature. All right, the next thing on the list is the evolutionary flow. So how does selection drive changes in the population? So there are three main types of selection, and you guys need to really know these well here. We have directional selection, disruptive or diversifying selection, as your note writer calls it, and then stabilizing selection. So I'm going to go through these real quick, and we'll talk about some examples of maybe how this would occur. And they give you some mice here, but we can try to think about some other examples that might take place. So directional selection. This is where you have a shift in or change in a trait in the population. So the book gives you an example of, let's say, I'll throw this out there. Let's say you have some type of small volcanic eruption, and then it cools and you have this volcanic rock. And so the mice that are there, which have a wide range of colors, the darker ones have found a place to hide in this volcanic rock. And because the darker ones are able to hide in this volcanic rock, then they're less likely to be preyed upon, and the population winds up getting smaller, darker over time because they are better camouflaged in that darker rock. Does that make sense? So that's an example of directional selection. Another one your book gives, and then we see this in the Galapagos Islands, is change in seed size. So in the wet years and dry years, we see a constant move back and forth to larger and smaller seeds. So seeds typically get, hold on, I can't remember. I want to say they get smaller in the wetter years for whatever reason. It's always backwards for what it is. Anyway, when there's more rainfall or less rainfall, the seed size is constantly changing, which then affects what else in the Galapagos? The finches. So the birds there and what they're eating. So depending on the finches, it's going to determine their beak size. If you've got lots more large seeds and harder seeds, then you're going to have birds. The birds with the bigger beaks are going to be the ones that survive. And so you'll see this movement in the bell curve of the population. In the disruptive or diversifying selection, this is where you kind of have a split in groups. So let's say you have a wide range of mice colors. So you have some light ones and some dark ones and some in between. Well, maybe you have this volcanic eruption and you have some, areas that are darker and the darker mice can hide there well. And then let's say that you have an area that maybe there's a hurricane that comes through and clears out some of the trees. And so a lot of this lighter sand is exposed. So the darker ones have a place that they can hide. The lighter ones have a place that they can hide. But the ones that are kind of in between, they're not really fitting in well in either kind of zone. And so they're more exposed and they get preyed upon more, and you wind up having more and more lighter and darker colored mice over time. And so eventually this disruptive or diversifying selection can lead to what? Your notes aren't shown on your phone, so put those up. What? It could lead to speciation. So speciation is the formation of a new species. So if you separate two individual groups like that, and let's say these darker mice are hanging out over by the volcanic rock and the lighter mice are hanging out over by the light sandy stuff, then if they're hanging out in those separate areas and they're just interbreeding with individuals in those areas over a long period of time, then that can wind up causing speciation, where basically they've just isolated themselves. So that is kind of a driving force behind creating new species a lot of times. And then the last one there is stabilizing selection, where you have individuals that get close, the bell curve gets closer and closer together. It's better to be like average, okay? So a good example of this is babies. So let's think of people having babies prior to like 100 years ago. If you wanted to have a baby, how big did you want it to be if it was like pre-1920s? Give me a poundage, ladies. Y'all are going to have the babies. You don't get to talk about this, Five.
How big?
Yeah, how big do you want your baby to be if you're having one before 1920?
Maybe about 4 or 5 pounds.
What's the problem with having one 4 or 5 pounds?
Well, they're really small.
Premature. And there's not a lot of preemie care, so your baby died. I'm sorry.
But then if it's really big, then
Well, how big?
you might die in childbirth.
Well, 6 or 7 is not really big. Like 7 or 8 is average.
Smaller than 4.
So normally average would be, and kind of fully developed, is probably going to be in that like 6.5 to 8 range. Okay? So if you have one that's much smaller than that, they're probably not going to survive or do well in the lack of prenatal care. And then if you have one like way over 8, then there's a chance that you can't push them out. You can't get them out, yeah. And so, or by the time you get it out, maybe it's dead. And so a lot of times, like back in the day, maybe like the baby dies or even the mother dies in some cases if they can't have the baby if it's too big. So the best sweet spot to be is like close to the middle, right? That 6.5 to kind of 8 pound range, where the baby's big enough to be born, but it's not, it's small enough that it can be born easily, but it's not so small that it has a hard time surviving. And so that's where you get into this stabilizing selection, where everything gets closer toward the middle, where it's better to have something in that similar range, close range. Okay?
And a question.
Yeah.
Like in the notes it says the bell curve. Like what would that be?
What's the bell curve? Okay, bell curve is something I thought y'all be familiar with. Bell curve is typically talked about when we're discussing like grades and stuff. So normally, and y'all haven't taken statistics, I don't think. So normally you have like a curve, and most things kind of fit in this middle portion of the curve, right? And so, like for example, a lot of you guys were in GATE or gifted growing up, right? And so normally the average IQ is at like 100. And then you have to be over here close to I don't know, you have to be close to like 125, 130, I think, to get into GATE. And so, and then you go down here to the other end, you're talking about down at 70. And so this is where people have like intellectual disabilities and issues. And this is where people are typically in like GATE or we call it gifted growing up. And so, but most people, the large majority of the population, I want to see within 2 standard deviations, you've got like from 85 to 115, you've got 60, it's either 64 or 68% of the population. So most everybody's falling within this group. You've got a few outliers on one end and you've got a few outliers on the other. And so the same thing if we're talking about like, you know, where babies are born. Most everybody's having babies in this like 6 to 8 pound range. You have a few that are really small, and you have a few that are like 11 pounds over here, and I don't know what the mom's doing, but anyway, crying for the rest of my life. But anyway, so, and then you might have some in the NICU over here that's like 1.5 pounds, that's barely surviving. But most individuals in the middle. So the bell curve is basically just a curve showing where most traits fall are close to the middle of that, okay? And if you take statistics later on, they talk about standard deviations and how far off they are. But most things, that bell curve is just kind of where the average, most individuals fall. All right, preservation. Genetic variation within a gene, within genetic, God bless America. Preservation of genetic variation within existing gene pools. Recessive alleles can be preserved as well as hidden. Now, the other day on your Hardy-Weinberg Teddy Graham lab, a question was posed to you, and basically the question was, and we'll think of it like this since you're eating the bears. Which trait is easier to get rid of, a dominant or recessive trait? Let's say you're a dog breeder, or you're breeding any kind of animal, and you're picking what traits get passed on. Which one's easier to get rid of, a dominant or recessive trait? Dominant. You see it, you're like, hey, little puppy, you're awesome, but you're not going to have any more babies, so I'm going to put you over here in this pen by yourself. So, and if you take and say like, well, I want to get rid of that recessive trait. Well, you might get rid of all the ones with the recessive traits, but then you have 2 of the dominant traits and they have some offspring, and then lo and behold, they've got a few of those that have the recessive trait. It's because that recessive trait is hidden, right? So it's hard to get rid of that because you can't see it. All right, the next thing, balanced polymorphism. So I'm going to give you the short of this here. This is talking about sickle cell. So, and one important thing you need to know is this term called heterozygous advantage. That means it is advantageous to be heterozygous, okay? So this comes up a lot on AP Bio. Now, it's an advantage if you're living in, just pretend you're living in like Central Africa. It's an advantage if you're a sickle cell carrier. Why?
Because you can be malaria prevention.
You have what to malaria? No. Resistance. This was on an FRQ one time, Mason, where they asked that question. And immunity means your body's fighting it off. So you already have like some innate way of fighting it off due to that, which you don't. I'm specifically talking about the sickle cell. The sickle cell makes you partially resistant because the malaria, the plasmodium, the protist that causes it, has trouble infecting those sickle cells. So you have a resistance to it developing, basically. So resistance is different than immunity, where you have an ability to fight it. Does that make sense? So, and if you didn't make that distinction on the test, then you didn't get the points. So I really want to make that with you guys. So remember, homozygous normal blood cells is good, but if you get exposed to malaria, it's more likely to infect and kill you. And then homozygous recessive, where you have sickle cell disease, is bad because of your ability to have normal blood flow and oxygenation and things going out through your body and normal just living because of those so many sickled cells. So both of those are worse than the heterozygous one, especially if you live in an area where there's malaria. All right, and let's talk about sexual dimorphism on 493 here. So di means 2. Now, I'm going to get in another thing here in a second where I'm going to tiptoe around this the best I can. So there are 2 versions, male and female, with secondary sex characteristics associated with either primarily estrogen or testosterone. So when we talk about sex, biological sex, we're talking about male and female. Now, back in the day, we did talk about, and I'm not going to get into much other political stuff here, but we talked about there's male, there's female, and then sometimes individuals don't have fully formed male or female parts. Maybe they have a little bit of both that started developing or some undeveloped portions of either. And a lot of times the reproductive tracts aren't fully formed of either one. But they're kind of like, they're not what we would say physiologically are fully female or fully male. And what are typically those people referred to?
Intersex.
Sometimes you're intersex if you have both. And a lot of times if there's this kind of in-between, not fully developed, a lot of times the term is intersex. So, but that's a very, very small portion of the population. I'll have to see what it is. But we're primarily referencing either male or female here. Now, normally in nature, you don't see this as much because, or if it happens, what if you have an individual that's like born either a hermaphrodite, normally when they should be either male or female, or they're where something is undeveloped, what would happen?
They're infertile, so they can't.
They're infertile, they're not going to pass on those traits, yeah, whatever caused that. So typically that's not passed on, and you don't see that so much. So let's go through and let's talk about this type of sexual selection that's taking place here. So intrasexual means within the same gender or within the same sex. So males fighting for reproductive rights. So think of like deer. They're fighting one another. Their horns are knocking. And that's a male deer right there. Yeah. And so they're fighting for the favor of a female, right? And then they get to pick the female, and then they go mate with her. And a lot of times the female doesn't have much choice in the matter, especially if you're talking about chickens. Man, chickens are violent. I had to separate my rooster. Did I tell you about that? He was getting a little too... Anyway. So, and then the other term is intersexual, means within 2 different things. So this is where you primarily have females, most of the time, that are picking the male. They are picking the male suitor. And a lot of times you see this with people where the female picks the male suitor for her to have offspring with, since she's the one having the offspring. Birds are a good example of this, Matthew. You ever seen the birds of paradise where they're like flocking around, they're doing all these dances, and the female picks like the best one? Y'all seen that? I got to show y'all the birds of paradise. And so they are trying to win favor, and the female picks them. That's an intersexual selection because it's between the 2 different sexes. Okay? And all of that is in effort to make sure the best genes get passed on to the offspring. All right. Any questions there?